Well, good morning, everyone. As I make some introductory remarks, I'll play this short film that is showing the implantation that we're leading up to study today. We completed the discussion of fertilization, and today we want to bring our discussion to the point of viewing this event slide to slide. But we know in reality it's a dynamic process. This still is a two-dimensional film, and this is not even the complete implantation. Let me see if I can call up another video really quick here. Oh, yeah. Let's see if we play this one. No, we're not going to get that one. Okay. But we know that the implantation is an ultimate endpoint. We will talk about the cellular division that you see occurring in front of you now, the changes as it approaches the endometrium, and of course the developing structures around it up to just about this point. This video, of course, is taking us through the entire series of development. I know it's rather small, but we will obviously proceed through the semester to understand and appreciate a vast majority of what's occurring through this entire sequence of events. You noticed almost a rotation in the orientation of things as they were proceeding through that series of developments, and we'll discuss that a little bit today. Very specific orientation, a polar arrangement of the embryo that develops, and specific orientation for implantation that is typical. So I know those weren't quite dazzling, but they at least set a background for the introduction of today's lecture. Just set my pointer here. Thus, subsequent to the union of the gametes through the process that we described, the fusion of the pronuclei, of course, mitotic division is going to begin, and this is the cleavage of the zygote. We'll follow that to implantation, discuss the formation of a bilaminar disc, the beginning of the embryo, and the extraembryonic structures. So at this point, I'll introduce this standard that in week 2, there are 2 layers of the concepsus, blastocyst as it's called at that stage. Week 3 is 3 layers of cells forming the embryo, and we proceed from there to the development of various appearances and morphometric changes of the embryo to begin to lay down the early organ systems. We will see with the implantation and formation of extraembryonic structures that part of the circulatory system will begin to form very early. So again, we had discussed the menstrual cycle with the recruitment and development of follicles, one mature follicle that ovulates and is drawn into the fallopian tube where fertilization can occur within the ampulla of the fallopian tube, and the remaining follicular structures, the theca and the granulosa cells differentiate into the corpus luteum that will degenerate if there is not a pregnancy and become the corpus albicans. So we start with the fertilization where we have the zygote with the pronuclei fused, and we'll discuss the cleavage process to the point of forming an intrauterine concepsus. Remember, concepsus is the term for this entire structure, where we will begin to see a vesicle forming inside this concepsus, and that is a cyst, and the cells contained within it and around it, actually around the cyst, are blast cells. So this is the blastocyst. They'll proceed to grow and enlarge and develop and ultimately bump into the endometrium and initiate implantation. We already see the orientation. The very typical spot for implantation is in the junction of the body and the fundus of the uterus on the posterior wall usually. And we see an orientation of these cells on the inside, that cluster of cells, inner cell mass, as we'll name it, identify it in a while. That will be the orientation for the attachment and the initiation of implantation, as we will see. Thus, as the ciliated simple columnar cells lining the fallopian tube are creating the movement from the peritoneal cavity out here toward the uterine cavity, the cleavage process, mitosis, is occurring at a rather steady pace, and therefore at different points in time we can predict how many cells will be in the concepsus. We also note that up to this point, just before or upon entry into the uterine cavity, the zona pellucida persists. It will be lost somewhere between this morula stage, as we'll describe, and the blastocyst stage. Excuse me. And when things go wrong, of course, implantation can be initiated anywhere along the length of the fallopian tube, or actually after fertilization might fall out into the peritoneal cavity and produce a peritoneal implantation, the ectopic pregnancies that we'll spend a bit of time discussing today as well. Thus, the cleavage is repeated mitotic divisions, which begin 30 hours after fertilization. So there's time required to get the pronuclei, the homologous chromosomes, paternal and maternal, have to come together, and then we initiate the entire mitotic division process, as typically described. So it takes a bit of time to lead up to that event, get all the machinery in place, fire up all the energy-producing reserves, get the cytoskeleton organized so that we can divide one cell into two cells into four. So the mitotic division, the nuclear division, will be coordinated with the cytokinesis, the cytoplasmic division, for typical mitotic division. It's not quite so typical, however, because there's no new cytoplasm produced for a long period of time, and the cytoplasm that existed in the ovum that was fertilized gets shared equally among all of these cells being generated through mitosis. So we'll see that as we proceed. These cells are going to be called blastomeres in the zygote up to the point, as I mentioned, for the blastocyst, and then the nomenclature will begin to change. So when we talk about embryonic stem cell research, we are really never looking at the embryo. It's always the zygote blastomere cells that will be dealt with. Well, not always just those. More frequently, or almost always, it will be those cells that we saw clustering inside the blastocyst called the inner cell mass. So the cells can be separated at various stages. As we know, if you separate the two cells after the first division, and those two each would go on to produce a complete embryo and fetus, you would have identical twins. So these are totipotent cells. That's why they are of such great interest in the research, basic biomedical research, and clinical research arenas. So at 12 to 15, there will be that solid mass of balls, solid ball of cells forming where there's about 12 to 15. And what's being implied here, of course, is that not every single one of the cells is going to be dividing at exactly the same period in time. We know that we will always have an end product when all the cells at any one stage have completed their division with an even number of cells. 2 become 4, 4 become 8, 8 becomes 16. And somewhere along the way, one might be lagging, and you would have maybe 15 cells. 12 also would not represent an equal division of all the cells within there. So strictly speaking, 16-cell stage is the morula. And actually, there's a distinct 16-cell stage, and then sometime after that, The 32 or 64 cell stage is when we begin to make the transition in terminology. So no one's at risk of being punished, at least here. If you're in med school, you might, if you're slightly off with that nomenclature and you refer to the 16 cell stage as the morula. More importantly, at that point, it's a cluster of cells greater than 16 that looks like a mulberry, and therefore the morula. That's the source of the term. So that's at about 3 days. So there's 2 cell populations that will begin to emerge as a cyst develops within this morula. So the morula is really going to be a series of cell development and division and overall morphological change within this concepsus, such that some of the cells are going to, well, let me pose the question first. What is contained within the blastocyst? Do you think it's air or liquid? Obviously liquid, right? So the liquid comes from somewhere. So somehow these blastomere cells are generating liquid in the extracellular fluid compartment within the concepsus, and it accumulates selectively in a certain area, and the cells will undergo intracellular communication and connection such that a cluster will stay at one pole and the fluid-filled cyst will be at the opposite pole. So we think of putting about 8 or 10 or 12 golf balls inside a basketball. Somehow they have to stick together and to the edge to be on one side, and then the rest of the inside of the basketball is filled with the fluid and is the cyst. So the blastomere cells have now started making some decisions on the overall structure and shape of the concepsus, and we have a fluid-filled compartment with a polar arrangement of an inner cluster of cells, the inner cell mass. This is the precursor of the embryo, and the outer layer will be the precursor of the supporting membrane. So we're going to proceed from that perspective. Well, as we saw in the video, 1 cell stage becomes 2 cell stage, 4 cell, 8, 16, so on and so forth. And along the way, somewhere after morula stage, the zona pellucida will begin to degenerate as the blastocyst is developing. You note also that the progression of these events is indicated with early blastocyst and later blastocyst. So we establish the general structure of the blastocyst in the early part, just after the late morula stage, right? Thinking of this as an ongoing transition. And that general structure continues to be instated in the developing and enlarging blastocyst. Mitosis is ongoing all the time. This, of course, is why any exposure to teratogens or any type of environmental toxin or whatever it might be, chemical exposure to radiation that can interfere with or damage the DNA replication process, cause mutation, can create a very high risk of malformation, maladaptation, or even failure to develop. Obviously, nourishment is really important. What is the mechanism for the movement of the molecules for nourishment and elimination of waste up until the point that we're still considering now? What's the biophysical mechanism to get the good stuff in and get the bad stuff out? Well, as it builds up, as the bad stuff builds up inside the cells and inside the cyst, what's it going to do? High concentration one area, low concentration another area. It's going to diffuse out, right? So diffusion, transport, passive, active transport, all those types of things to move good stuff in and bad stuff out. Except for lipid-soluble molecules that have free diffusion, the water, virtually all the other molecules will depend upon membrane proteins for movement, as we've always discussed with cell membranes. So ionic maintenance, everything. So there must be something in the fallopian tube sustaining this. There must be something in the endometrial, in the uterine cavity lining the endometrial surface that supports this. What is the source of the nutritive material lining the endometrial surface. What stage of the menstrual cycle are we in with reference to the uterine cycle? Secretory phase. After ovulation, a secretory phase. So here we are, 3, 4, 5, 6 days later, still a lot of secretion going on. Okay? So by the time we have the blastocyst approaching implantation, there is a diffusion of those nutrients away from the uterine lining, and it's not like the uterine cavity is, all inflated and filled with air or gigantic, fluid sac or something. It's really quite collapsed upon itself with very little intraluminal space. So we're not talking about, 10 feet of diffusion. It's just maybe a few millimeters at most for these things to be diffusing to the cell and away to the conceptus and away. So we see that the continued instatement already, we're showing a 2-cell layer here, and we obviously look at these at higher magnification as we proceed. And then these outer layer of cells become very well defined. You see that they look like fried eggs. It's a simple squamous epithelial lining. And we have the inner cell mass surrounded by these cells out here called the trophoblast cells. We'll look at these again and again and follow their development. And the zona pellucida disappears so that we no longer have this constrictive eggshell kind of a thing surrounding it, and it can begin to enlarge. Enlarging not only by increase in cell number, but by an increase in fluid in the blastocyst. So I remind you that there are illustrations in black and white that are very simple to post for exams, so you should be able to identify structures in slides such as this and predict, what's the morula stage and obviously 2-cell, 4-cell stage, and answer indirect questions such as if an arrow was pointing to this structure here and you were asked to identify that structure, what would you call it? What is this little indentation that forms on the outside of the cells when the cytoskeleton acts like a purse string for cleavage? The cleavage furrow. So classic mitotic terminology. Other series of events are occurring, as I had stated, as we proceed from an early 8-cell stage. What's missing from this illustration here at the 8-cell stage? Zona pellucida. So it is there. It contains the structure, but it's just removed for the ease of illustration, or the artist just didn't draw it in the first place, didn't have to remove it if they didn't draw it, right? And then at the later 8-cell stage, there's a term called compaction for what occurs, and this is, as I had indicated, the cells are next to each other. They begin to insert, or they already have, proteins in their membrane. They're sticking to each other, talking to each other in that sense, by cell-to-cell contact, and they form tight junctions to hold themselves together so that they don't separate. So what this tells us is that as mitosis is occurring, we do get a bit of an interphase in there because the cells are expressing genes somewhere along the way and kind of changing what's occurring. So they send the internal messages, whatever they are, by communicating with each other. Hey, let's get together and form a tight junction here. That takes coordinated effort among and between cells. And then the 16-cell stage, there really is an orientation of an outer layer of cells and an inner layer of cells that, of course, will proceed to develop into the blastocyst with the outermost cells, the peripheral cells. And again, this is just showing a two-dimensional view. These whiter cells would completely surround it like the basketball, and then the golf balls would be all inside. And then ultimately the collection of the fluid within the blastocyst, the cavity, the blastocele is the term for that cavity, C-O-E-L, blastocele. The fluid building up in there pushes the inner cell mass to one side, and they're all stuck together by tight junctions, as I use the metaphor to the basketball filled with the golf balls. They're not all the golf balls won't end up in one place unless they're stuck together in the very beginning. So the formation of these tight junctions at the compaction phase in the late 8-cell stage is critical for all of the cells of the inner cell mass to stay together and be pushed to one pole inside our basketball here. So the trophoblast cells will become part of the placenta, and the inner cell mass will proceed to develop into the embryo. So the blastocyst, that ball of cells with the fluid-filled cavity, the blastocele. The trophoblast is the thin outer cell layer that's going to contribute to the placenta. It's not going to become the entire placenta, it's going to contribute to it. So obviously more cells are going to be needed. We're going to have a lot more mitotic division, a lot more differentiation. Remember, we're at the point now where we only truly have totipotency in the inner cell mass cells. And this is a stage where it's large enough to be viewed under microscopy in petri dish or something like that, and manipulated, can be grasped with micro manipulation devices. The trophoblast cells can be penetrated with a micro pipette, and these cells can actually be withdrawn from inside the blastocyst, the blastocele, and removed, and each of these is actually totipotent. Maybe not totally successful in producing a complete embryo, fetus, and human being. There's a little bit lost along the way as things proceed. What is it that these cells cannot produce if you were to take one and try to grow it into an embryo or a fetus? Is there any evidence of determined differentiation directions at this point? We got two colors, so it should be kind of obvious, right? These cells, the cell mass blastomeres, cannot produce trophoblast cells. So if you're going to take more of these and try to generate another blastocyst, you have to have a donor or host blastocyst in order to inject it within there. Sometimes these are removed, so you have the hollow blastocyst. Other times they'll just inject the inner cell mass from one blastocyst, and somehow it takes over, or you end up with a mosaic even sometimes. So all different approaches obviously have been attempted. This image here is showing us that there are detailed illustrations. Again, this is from the Gray's Anatomy. A lot of black and white illustrations, halftones. And we see that what I like about this one is it's showing us that these are strings of cells all distributed around here. So there has to be extracellular matrix holding them in place. So somebody's making extracellular matrix. We don't have the eggshell to hold things in place anymore. So these cells are doing a little bit of work as they're proceeding through their mitotic division. And somewhere within here we've got to have cells that are rapidly undergoing mitosis, that probably the fastest that rate that could occur. If there are, I don't know if there are different rates of mitotic division or if there's only one. So if there is only one rate of mitotic division, assuming all conditions are identical, then the only way to have greater numbers of cells produced is to have a greater number of cells with mitotic capability. So the inner cell mass is shown here. So it's not just this simple two layers of cells or small cluster of cells. It's actually numerous cells. Or we can simplify this to the point of having this type of illustration with arrows labeling structures, and obviously I provide you the key to this illustration, and it's an obvious performance assessment tool that you can expect to see. So we've talked about the inner cell mass, the blastocele, or blastocyst cavity, and the trophoblast cells. So this simplifies it tremendously, but this may be the way that it actually appears in the early blastocyst stage. The previous slide, maybe that was a little bit farther along where there's a few more cells in there. And then obviously as we proceed to the 2-cell layer, embryonic, disc, it would be different. So here's from the textbook actual histological samples of the human embryo. The very early blastocyst stage, what's this structure here? Zona pellucida, degenerating zona pellucida, so it's not continuous all the way around. Remember, this is two-dimensional. It's a cross-section through. But we do see that a cluster of cells are staying together. Because of compaction at the 8-cell stage, they have tight junctions and are sticking together. And we're beginning to get the collection of fluid in this area, extracellular fluid, the early part of the blastocyst cavity. And then a little bit later, when the shape is well established and the inner cell mass doesn't quite have as many cells as we saw in the illustration by Gray's Anatomy. Let's continue then to envision this developing blastocyst. Remember, it's not always just like this or just like this. It's always freeze frame and is constantly ongoing over hours and days. By day 6, there will be, this is the typical, remember, it's never exact in biology, but relatively 6 days after fertilization, we can expect a high probability of the attachment of the blastocyst to the endometrial lining. So where are we in the menstrual cycle now? Is it pre-ovulatory or post-ovulatory phase? Obviously post-ovulatory phase. Are we in follicular phase or luteal phase? Luteal phase. And are we in proliferative or secretory phase of the uterine cycle? Secretory phase. Okay, so we've established it's post-ovulatory. Roughly where are we in that post-ovulatory phase? Or luteal phase or secretory phase? How long is that post-ovulatory phase? Basically 14 days is a good reliable number. So we're about halfway through that phase. So the corpus luteum is just about tightening the noose around its own neck to begin the separation from or development of independence from maternal pituitary. So the cells that we've been looking at have to begin another function. What is that function? They take on an endocrine function and begin expressing the genes for human chorionic gonadotropin and secreting that. So, we're inside the uterus. Where's the corpus luteum? In the ovary. So we've got a pretty tremendous task of getting HCG from the intrauterine cavity into the plasma, to the heart, through the venous system, through the heart, through the lungs, back to the heart, out through the aorta, down finally through the ovarian artery, and finally in the capillary beds delivered to the corpus luteum. Seems almost impossible, doesn't it? What makes that a possibility? We already stated that this intrauterine environment at this stage is primed for what biophysical property for molecular movement? Diffusion. When we talked about the development of the endometrium, the proliferation, we talked about the epithelial cells, the decidual cells, glandular cells, and what else? What other? Vasculature, right? You can't absorb and wash away into the venous system without the vasculature. So the environment is there, but we've got a protein that somehow must be protected so it's not degraded. All hormones can be acted on to be degraded. So, again, it's always so easy to just think, oh yeah, HCG keeps corpus luteum going, we got pregnancy. When we saddle up our molecule and follow it from the cell of origin to the target cell, it actually becomes quite amazing at times. So there is marvel to all of this simplicity that we seem to just stream right by. Thus, the trophoblast is going to, so on the embryonic pole, we had indicated that's the pole that typically attaches to the endometrium, somewhere around day 6. The trophoblast is going to begin to differentiate into 2 layers. The cytotrophoblast, the thinner layer of cells that maintains the margin of the blastocyst, so the outermost portion. The syncytiotrophoblast, an additional layer found only at the embryonic pole, extends into the maternal endometrium. So again, from that video that we saw, there was the polar attachment at the end, the embryonic pole where the inner cell mass was located, and the trophoblast cells that were at that edge of the basketball are actually different than the rest of the cells forming the basketball, and they are become the syncytiotrophoblasts that are going to divide rapidly and infiltrate through the epithelial lining of the uterus and into the stroma of the uterus. What's the difference between stroma and parenchyma? Are the decidual cells within the functional layer of the endometrium parenchymal cells or stromal cells? Parenchymal is functional, right? We're the parenchyma. I remember doing this just the other day. All you people in here, you're the functional cells within this building. You're the parenchymal cells. The stroma is the whole building that contains us, the structure that supports us, right? Chairs, tables, walls, things hanging down from the connective tissue fibers hanging from the ceiling there. Okay, so the term is that the syncytiotrophoblasts infiltrate the stroma of the functional layer of the endometrium. And just so happens the decidual cells are right there too, so very convenient. These decidual cells are, as we had said, through the secretory phase of the uterine cycle, they're loading up with stored lipids, stored glucose, and whatever is there to store the amino acids for protein synthesis. So ultimately we'll see there's a decidual reaction where they just go, and then the invading syncytioblast cells that are differentiating will be bathed with all of these nutritive materials. So this takes care of week 1 of development. So at the end of week 1, we have the blastocyst, somewhat late blastocyst. Implantation is occurring, at least as attachment, with the beginnings of the differentiation of the trophoblast cells into cytotrophoblast and syncytiotrophoblast cells in a polar arrangement, although the syncytiotrophoblasts are more restricted just to the embryonic pole, and they're going to undergo massive mitotic division and migration. It's more of a migration of just a clump of cells just dividing and physically displacing the cells that were on the surface in the first place. So they don't break off and wander away. They move together as a solid core of cells, and we will see that. So around day 6, the blastocyst would be attached to the endometrial epithelial cells, and the inner cell mass is going to begin to shift a little bit, and the cells are, remember, we're also getting more intrablastocele pressure, so it's going to keep these cells just from bulging and going, taking over the blastocele. So they'll just start to form 2 layers, roughly 2 layers. And this will continue to develop as the syncytiotrophoblast cells infiltrate the endometrial lining. So we'll see more of that at higher magnification as we proceed. The syncytiotrophoblast, as I said, erodes the endometrium. I don't really like that term. the erosion that we're more familiar with is the potholes we're going to see in the road after this horrendous weather. Right? We all know the roads where we hit them, and the potholes are just going to be much worse. So it's not really that type of an erosion. I don't think that it's corroding away cells and big chunks of the endometrium are falling off. It's just simply secreting molecules to loosen up the junctions between cells, and then you just get a couple cells slip in there and then form a core of cells, and suddenly you're moving in. And we saw that in the video, however small or large. So now, of course, this syncytial trophoblast, about day 7, 8, 9, so we're progressing toward 3/4 of the way through the luteal phase, where the corpus luteum is pretty much instating the condition of independence from maternal pituitary gonadotrophins. So the maternal FSH and LH levels will begin to drop just as they would during a cycle without pregnancy, right? The HCG steps in to sustain the corpus luteum and the levels of progesterone. So here's where I'd reminded myself to play the implantation animation. We already saw that at the beginning. I don't want to try to fumble to find it and do all that stuff. So we can just quickly take an overview look at this illustration. Again, as we proceed, we'll see everything again. We kind of go through things once, and then we go through things again, and then we go through the organ systems, and we kind of remind ourselves of everything else that's around. So I know these are small images. You have them in your textbook and in the PowerPoint or PDF slides that you can print out. I think I got just about all the PDF slides for what we need right now loaded, so you can print them one slide per page. So I know you can at a later time see these at a larger stage. So we are seeing this rather massive infiltration by the syncytial trophoblast cells, and actually will begin to draw the entire blastocyst into the functional layer of the endometrium. Obviously, along the way, going to encounter glands, obviously the decidual cells, but the glands and the remember those venous lakes that we described? So the highly vascularized endometrium, will be present there as well. Does the vascular system become eroded? I'll use that term. Yeah, ultimately it does, right? Humans have a hemochorionic type of placenta, so blood onto the surface of the chorion. So there would be maternal lakes that will actually form within here, and we'll see that as we proceed. So what we want to do is begin to take a look at really what's the inside of these little projectile columns of cells of these syncytial trophoblast cells. There's other changes occurring with the those cells of the blastomere cells of the inner cell mass, the 2-cell layer. We see now that it's actually beginning to form a second cavity, so let's proceed to see what we have on that as we obviously that'll come through the next series of lectures as well as we get through week 2, week 3, and so on and so forth. This is the point where we interject the discussion of ectopic implantation, because we've talked about implantation. So it doesn't always have to happen just the way that we described. The vast majority of the time, obviously, it does, but something might happen. We don't really know what it would be. What could be a physical condition in female internal reproductive organs that could increase the risk of tubal pregnancy? I know Hank Hill's got a narrow urethra, but that wouldn't be what we're talking about here, right? So, well, I'm thinking more of just a narrow fallopian tube for whatever reason. Could be scarred from sexually transmitted diseases, STDs, yeah. Haven't taught the human sex and reproduction class in a long time. Or it may just be, for some reason, the isthmus is smaller than usual, or a variety of things like that. I guess what I could picture for the endometriosis as causing it is if the endometriosis had impacted the fallopian tube. Endometriosis usually is endometrial cells within the peritoneal cavity on the surface of the body wall or organs that ultimately can cause some localized development of connective tissue, kind of scarring, and things like that. So it couldn't be so restricted in its lumen that the sperm could not penetrate, but obviously, as we know, the sperm are a lot smaller in diameter than the ovum or the zygote. So anywhere along the way, throughout the oviduct, within the abdomen, on the ovary, on the inferior uterus, any place is a target. So this slide is showing us an illustration on the left and an actual pathological, gross morphological photograph on the right-hand side. There's a ruler here. These must, of course, be centimeters or centimeters. I did some training in pathology in Canada, Montreal, and everything was a centimeter there. So what we see, we're spanning from about 1 to 7, so it's about 6 centimeters long. And we see that in this case it was an implantation, a ectopic tubal implantation in the infundibular ampulla junction. So there's not much more room for this to grow. Obviously, this had to come out. It was, I'm sure, extremely excruciatingly painful. If it had ruptured, there's a risk of hemorrhage. There's a risk of infection. Why is there a risk of infection anytime we're talking about the fallopian tubes? It's continuous with the external environment. Vaginal orifice, cervix, uterine cavity, fallopian tube. So you get something like this causing an irritation. Any amount of bacteria that may be present or could have found its way in obviously would have a great time with this necrosing material. But simply, this is painful, and then if it did begin to necrose, obviously, that would be even more painful. Yes?
In the book, it said that abdominal ectopic implantation can be carried to full term, but yes. Where does it get its nutrition or its supporting membrane? Or how would it not be? I think we've got a slide
for that. Obviously, this cannot go to full term. And anywhere along, as I said, the highest risk would be the implantation occurred within the intramural portion, so right within the wall of the uterus. Extreme risk of hemorrhage there. So there's a wide range of locations. As we said right here, the X, the posterior wall of the junction of the body with the fundus is, the A number 1 site. But anywhere along the way, if it happens to, after fertilization, as I said, fall back out of the fallopian tube, virtually any location in the peritoneal cavity. And here we see in letter G that it has become attached to the mesentery of the small intestine here. The mesentery is a neurovascular bundle, basically, and it is very highly vascularized. Any place with a capillary bed that is approachable within just a few cell layers is a candidate for this. And within the peritoneal cavity, that's basically anywhere, because there's the peritoneal lining, which is the same as the perimetrium. It's just peritoneum draped over the outside of the uterus. It's a single cell layer thick, so that's not much. And it's even flat cells, not the cuboidal cells of the uterine lining. So these syncytiotrophoblasts will have no problem working their way through that cell layer. And just underneath, you've got connective tissue. Connective tissue is always highly vascularized with a rich capillary bed. And as soon as it establishes that, proceeds as if it's the uterus. It's not exactly the same, right?
So, it also said in the book that it gets most of its nutrition from, like, use of the uterus and all the secretory secretions from it. So if it goes the opposite direction, and you have implantation in the tube or in the abdomen, does not being close to the cell, the secretory cells, does that affect the development or?
Typically, by the time, you know, I'm not a clinician, so I'm not going to pretend that I am. But typically, by the time that a tubal pregnancy comes to your attention, that isn't such a great concern. And I don't think that there are any cases of tubal pregnancies that have been saved. You know, if they're detected very early, they can be removed, you know, by inserting the appropriate devices to try to wash it out and withdraw it. Obviously, there's a risk of scarring after that, but the typical case, you know, gynecological surgeons just go in and cut it out. That's their approach. So in this case, there's about a 4-week embryo. And in just taking a quick look at it, everything seems to be all right. So somehow, up to this point, it can get what it needs. We know it's not exactly perfect, but if you've ever seen, either with surgery or in cadaveric specimens, the abdominal cavity, and you try to picture a placenta in there as big as they can get, you know, it's just going after a tremendous vasculature, so it can't be too fun. Push the button so everybody. Push the button. You got to hold it. Yeah.
So the people don't come.
Because some people don't come to class. They're taking.
Oh, okay. I've seen several ectopics just through when I was doing ultrasound. Most of the time, it's not a viable fetus. By the time we get it, it's usually just a mass. It's not a viable fetus anymore. It's usually just a large mass.
Even intraperitoneal, or you're saying the other?
Yes, anywhere, either intraperitoneal or within the tube. It's usually not a viable fetus. It's usually just a mass. The fetus is already demised, and it's usually not a viable fetus. There have been a couple of cases. One in particular that really stands out in my mind, but I won't go into details. But usually, by then, it's not even viable.
Yeah, and this is, I think, a prime example of how to make a point of emphasizing the, you know, basically 100% failure of any pregnancy other than within the, anywhere associated with the uterus other than within the uterine cavity. So if it lands intraperitoneal, what if it landed on the outside of the uterus and somehow got in the other direction? Maybe it could thrive and survive. But it's not 100%. It's just there are occasions where a peritoneal can go to full term. Yes, it's one of those faulty approaches of reminding people of their anatomy and what's the difference and what's the clinical correlation to that. Right? So an implantation such as this would have a better chance of proceeding to full term than one within the tube as we had seen. So this is a sagittal view of the question. Yes, go ahead.
How common is this? Like, do you know, like, the percentage of women, like, is it just real common, or?
I don't think it's very common at all.
Okay.
Now, it's not extremely rare, tubal pregnancies at least, but I really don't know whether it's 10 in 1,000 or 10 in 1,000,000 or really what the percentage is. So in this view then, of course, we are seeing a sagittal view of the internal reproductive organs of the human female, the urinary bladder here, the uterus here, the ovary and the fallopian tubes. So after fertilization, the zygote fell out into the peritoneal cavity and attached to the lower part of the cervix. and the rectum. So I doubt that this one could be successful. You've got to poop at some time. Okay, we'll stop here and pick up with this discussion on Friday.